All right, good evening, everyone. This is Dr. Prasanna, and I thank Barcode Biotechnology for inviting me as a resource person for this webinar on current trends in next-gen DNA sequence analysis or sequencing. Okay, so today what we will be seeing is what are the trends in the next-gen sequence analysis, various platform available, what is the current trend among these platforms, how to analyze the next-gen sequences. In order to understand all those things, we need to realize why we need next-gen sequencing and what are the fields these next-gen sequences are applied. Okay, so today, before getting into the topic, I want to introduce who is Barcode Biotechnology. Barcode Biotechnology, founded in 2013, is a specialized DNA sequencing analysis company. Um, we are, I can say, most affordable sequence service provider in India. We have branches headquarters in Panji, Goa, and we have a centers in Bangalore, Chennai, and also in Telangana. So each and every branch have their own specializations. So sequencing with regards to next gen, it has been solely focused in Goa centers, and we have Bangalore dedicated for Sanger sequencing, and Chennai office is for mostly for aquatic testing using DNA sequencing, and Telangana is right now acting as a pooling center focusing on more samples towards the East India. All right. So what we do is our infrastructure consists of all necessity instrumentation and technologies required for DNA sequencing. Uh, for example, we have a Mermaid 192X synthesizer, a primer synthesizer, which runs 24/7. I would say they are the bread supplier for the entire company. We have Megabase. We started with the Sanger sequencing in Megabase. We have a ABI sequencer 3700 that are highly involved in Sanger sequencing. We have a couple of PCR workstations, and we have Illumina HiSec MySec. To be fair, we are actually for in case of NextSec or NovaSec or long length DNA sequencing, we are outsourcing the samples. So HiSec MySec we are doing it in Panjim, Goa, and next sec, we are outsourcing the sample to Macrogen, South Korea. All right. So this is we, and these are various services we are providing. We are, as I said, most affordable sequencing service provider in India. You can simply send us even mixed cultures for microbial identification, point out which bacteria you want to identify. We are also working with Archaea identification, mushrooms. You can simply send us a cube of mushroom sample whose ITS gene is being focused for species identification. In case of multilocus gene sequencing, you can point out any genes you want. In case of new species discovery, most of the time for bacteria, you need to sequence both 6NS rRNA and RPOB or the whole genome of a bacteria to prove them as a new species. So multilocus, we are doing even phage identification, phage plaques you can send to us. We are doing phage sequencing services, animal barcoding where cytochrome C oxidase subunit 1 gene will be focused for identifying any animals, including terrestrial, marine, even community samples. We are accepting for meta barcoding, where we simply extract DNA from the community of an organism. We can use HiSec, MySec to decode it. Right now the sequencing market is all about 14,000 to 15,000 rupees for next gen sequences. If that is too much, we can still go for traditional cloning sequencing. If you are targeting a presence of particular species in a community, you can simply tell us or give a specific primer where only we will amplify that particular species from a community. Go for cloning and sequencing. We are not charging anything for cloning and sequencing. Yeah, per sequences cost here is 1,850. So instead of, if you want to detect some species in a community, instead of Illumina platforms, you can still go for traditional cloning and sequencing. Using Sanger platforms, we can provide you these services. So we are closely working with the central and state government forest departments during various census and surveys. We usually camp with them, collect fecal samples. We can decode the entire animal biodiversity or animal dietary composition, what they are feeding on, and the animal species from their fecal samples. Even when birds, when there are many red listed species where you cannot sample them up, you cannot collect samples by toe pricking or any other method. So in that case, we depend on the fecal sample for bird species identification. Okay, many birds shed their epithelial cells along with the fecal samples. That dried fecal samples can be collected, and there we can use cytochrome C oxidase subunit 1 mitochondrial gene for bird species identification. Plant authentication we are targeting RBCL gene for angiosperms and MADK for gymnosperms. Not only plant tissues, you can even send us herbal powders and we can decode the entire biodiversity of plant species that is being used for the herbal for powder authentication. Recently, FSSA and many ISO organization demands meta barcoding data or meta DNA sequencing data for herbal powder authentication. Okay, so we are also doing giving that service. We are also giving something called eDNA, yeah, non-invasive sampling service. Provided you want to decode many of our customers, we have given this service, especially for marine ecosystem or in pond water samples. If you just send us a filter paper that has filtered 1 liter of the sample, we can simply extract the DNA, use Illumina platforms to decode the entire biodiversity in the aquatic ecosystem. Whether from microorganism to macrofishes, we can simply decode the biodiversity using the next-gen sequencing platforms. So these are the various services actually we are providing here. So this let's get into what is this next generation sequencing. It will be highly unfair if we do not talk about a important project in human history, that is human genome sequencing, which is a catalyst to enhance the various technological platform in next gen sequencing. Okay, imagine human genome sequencing. It took 20 years, $3 billion, completed by 2021, used a Sanger machine for creating a genome. Okay, see, at that time, 20 years back, it is around $100 million per genome. So imagine the cost of the sequencing. Not only that, it will take more time to, for the sequencing run. But this human genome project that fueled these various sequencing technologies starting from 1975, where Sanger Sequencer is very famous platform for sequencing the any genome. And by around 1990, you had the first generation by Sanger, followed by the second generation sequencer, which was introduced a massive parallel sequencing with the platforms like Illumina and Torrent. This enable high throughput sequencing. This is where it has started. And then currently we are at third generation where PacBio and Nanopore, they are offering long read and single molecule sequencing capability. We will see why long read and single molecule sequencing capability is very important. You see, Sanger, what was the disadvantage is Sanger can handle up to 384 samples at a time. They can produce around 700 base per reads on average. Okay, if you have a fresh laser machine, I know they can produce much more longer, but 700 base per reads. Okay, they can produce up to 1 million bases per day. You may think 1 million is very big, but it's not. Okay, human genome is around 6 billion bases. Where is 1 million and 6 billion? So for to cover every genome, you may have to sample them 7 times. Okay, so if you go with this speed or this numerical value, it would take around 100 years to sequence a human genome in one machine. So how did this human genome project happen within 20 years? They ran a Sanger sequencing factory. Okay, every sample was split and simultaneously it was run in hundreds of machines. And that's how they were able to complete that in 20 years. But today we can do it in 2 days. I will tell you how. Okay. So before getting into the current trends, let's see how we evolved into the current trends. Initially, it was a pyro sequencing platform that has produced 400 to 700,000 base per rates. Then we had Ion Torrent, Illumina. Right now, Illumina is a dominant player in the market. Okay, they work by sequence by synthesis. And those Illumina platforms are followed by solid platforms, DNA nanoball sequencing, Helicos, PacBio, and finally we are into third generation, which has longer reads up to 25,000 to 30,000 reads at one sequencing reaction. Okay, so see, main important thing about if you ask me what made a particular platform to thrive and particular platform to go off the grid is the error rate in the sequencing data. Okay, Nanopore still suffers from this. They have up to 15% error rate, meaning out of 100 base pair you sequence, maybe 15 base pair could be a error. But in Illumina, it is less than 1% error rate, meaning at least one nucleotide might be error among 1,000 nucleotides you sequence. So that's why the Illumina platform still survives. So if you want to put this tabular column into a graphical data, so the read length of 454, Illumina, Ion Torrent, and PacBio is shown. You can see that shorter the reads, usually the yield will be more high. The throughput per run in gigabases, you'll see that will be of high values. Okay. If you compare last 25 years or last 15 years from 2001 to 2015, you can see the cost of human genome has been consistently decreasing because of evolution of technology, and reagent cost has been brought down. Brought down. All right. So this is runtime per in every platform, and for sequencing a whole genome of a particular organism. If you want to put everything in one slide, you can see, you can compare the various platforms here. At Barcode Biotechnology, we have MySec and HiSec, and NextSec, we are actually outsourcing to Macrogen South Korea. Okay. So how this sampling is done and how your samples are treated. Every sequencing reaction will start with first step, that is DNA extraction, high quality DNA extraction. Okay, these extracted sequences, DNA samples were actually fragmented, and they may be ligated or captured. The library can be prepared based on your objective, and you have to decide whether you are looking for a short reads or long reads. See, for microbiome, even though MySec is most probably used, that can yield an average of 300 base pairs. But you see, the molecular gene, a taxonomical gene for bacterial identification is 6' rRNA, which is 1,500 base pair. Many times we are seeing this. You may be focusing on V1, V4 region. Why these regions came? V1, V2, V3, up to V9 we have. Why can't we use 1,500 as such? Why to have V1 to V9 reach around 150 to 200 base pair is because of the sequencing technology, because of the limitation of sequencing technology that cannot read the entire 1,500 base pair. Even Sanger's read an average of 700 to 800 base pair. That's why every time you identify a bacteria, you need to go for two-way sequencing, where 900 plus 900, you can compile both forward and reverse strands and produce 1,500 base pair. I have seen whenever the sequence length of 6' or a gene that goes down below 1,200 base pair, the bacteria will have 100% similarity during blast analysis with more than 7 species of bacteria. Then what's the use of going for sequencing if they cannot propose one particular species out of blast analysis? So that's why it's very important to choose the right sequencing platform. If taxonomy is your target, you have to think which platform is the right platform for you. Do you need a PCR? These types, even PacBio Nanopore, they don't need your PCR. Just extract the DNA. You can go and sequence everything out. Whatever sample you have extracted, you can sequence it through various sequence analyzing platforms. You can generate the whole genome or extract only taxonomic data out of it. So various platforms records various ways of preparing the samples. So why this platform and how they are useful to us? These platforms find application is in various research, diagnostics, and healthcare. Okay, one of the main important point in NGS sequencing is now why everyone cannot afford it is not only its prices, which I feel I'm seeing this in last 10 years in India. Initially, around 2000, it was costing in lakhs. Okay, right now in a market for a Illumina HiSec or MySec, you can do it around 15 to 18,000 per sample. Okay, why always a price range? Why not a fixed range? Because the customer has to tell the company what is the sequencing depth, how much million base pair they need, what is the target of the genome, and most importantly, after creating the raw data, what kind of analysis they require? Do they require an analysis? If yes, taxonomic analysis, or if you are working in multi-drug resistant genes, what are the genes you want the company to bin out? and give it to you. Okay. To be frank, I am seeing many customers spending 15,000 per sample for my high-sec, MySec, again spending 15,000 for analysis. Okay. So I would say this sequencing analysis is actually 30 to 40,000 per sample, which many companies will not be declaring initially, but once you created a data out of 15,000, you will see you need a proper bioinformatician. Even though many cutting edge AI tools are right now available, still you need a human optimized pipeline even for AI to pick your raw data and give a meaningful results. Okay, so DNA sequence analysis, especially Sanger sequence, Illumina's next gen sequence analysis is a big market. I really feel in next it's already happening, but in near future too, they have a high value because these technologies find their expression in genomics, transcriptomics, epigenomics, metagenomics, whatnot. Through various other individual assays, you can see the NGS sequencing dominating the market. Okay, so if you are a first-time customer, I would always advise you. There are various approaches you can use for your genome analysis, where you can apply your NGS technologies for data generation, data analysis. Whole genome sequencing and whole exome sequencing, internal transcript spacer, especially if you are working on fungal biodiversity, and chip technology, the chromatin immunoprecipitation technology, and assays for transposis accessible chromatin or antimicrobial resistance. There are various ways you can actually choose an NGS platform. So if you are a first-time customer, first-time student or scholar or a employee is going to use this next-gen sequencing, I would say you should be very clear about your objective. Why are you going for this platform? What is the research question you are trying to answer through NGS sequencing? So then you should choose your sequencing platform. Many customers, they come up with an idea. They say, I want to do this. Can you propose a sequencing platform, sequencing, million, how much depth of data has to be produced, what kind of primer to be chosen? I'm telling you, you are asking for trouble. Do not ask a company to choose a platform. They will definitely go for their convenient way of creating a data and giving to you. Okay, so whenever a customer asks for a sequencing platform, company knows that you are a newbie in this. Okay, please don't do that mistake. Read some research article in your domain. Okay, or if there is no article working in your domain, parallel articles, used the parallel sample similar to your samples. Understand their methodology. Why did they choose a particular technology platform, and why did they produce this data? Okay, you understand. If it is, say, for example, microbiome diversity work, you should know how many millions of reads will be sufficient to capture entire diversity in the sample. Okay, say for example, if it is an extreme environment like a deep sea ecosystem where you may be, deep sea mount, if you are thermophilic mount, if you are going and sampling out of there, the diversity expected is very minimal. So 5 million to 10 million reads of next gen sequences will be sufficient to decode the diversity. But if you are going for a garden soil sample or any other soil sample where the life activity is more, you are expecting more diversity. So you may require 15 million to 20 million reads to capture the diversity. And whenever say microbiome diversity, what group you are most interested in? If it is a normal proteobacterial group, what kind of region you should be targeting? V1 to V4 or what else? If it's an actinobacterial group, or if do you want V9 region? So you should choose which region to look for so that your objective is clearly met. Do not ask the company to propose a primary region. Definitely, they are there for business and they will propose what it is convenient for them. Okay, an analytical pipeline. I'm very sure that most companies will charge you the same amount for creating the data from your sample. They know this. Okay, whenever they created the data, they know the customers will come back for sequence analysis, which is time consuming and skill required process. Okay, if I will show you DNA sequence analysis, especially next gen NGS sequence analysis, is a big market by themselves. Because see, there are various panels you need to decide while preparing a library for loading your sample. based on the various objectives you have. Okay, and what kind of sample you should be taking for that. Okay, so if you clearly tell the company, this is the platform I want, this is the gene I'm going to target, and this is the basic bioinformatics analysis I need. Only when you know from A to Z, you can get a clear and cheapest quote from any NGS DNA sequencing service providers. Okay, so basically, when they receive your sample, they will just go for fragmentation, build their library construction, out of which the target primer will be used for amplicon selection, or if there is no PCR requirement in case of Novasec, you can directly go for whole genome and sequence out every sample there. Okay, so here is a common analysis of various sequence analysis softwares available in the current market. Okay, there are hundreds of softwares. This data is compiled in a publication by Hina et al. Last year. So I thought I will show. In barcode biotechnology, we have a separate bioinformatics division, barcode bioinformatics, that they are focusing only on this pipeline optimization and sequence analysis. Okay, for simple cleaning and for aligning the sequences, we are using FastQC, FastX2, it's multi-QC. For whole genome and exome analysis, we are using ANOVA or Picard. So the order of the tools I have shown here is actually based on the priority. Okay, so transcriptomics, epigenomics, methyl sequencing, or chip sequencing, the tools are different. Okay, that's why the DNA sequence analysis softwares are, they are different world by this itself. They are billion-dollar industry, analyzing, using them for uh, deriving a meaningful data out of NGS sequences. So 6NS rRNA came to, if at all you are studying a taxonomy with a large data and you want a faster result, if you have a minimal, relatively smaller data, and you can spend more time, Mothur is the right tool, usage, green genes. There are various platforms that can be used for sequence analysis. That's why you see the market. Whenever right now the DNA sequencing and next gen sequencing market is around $15 billion last year. This year estimate will come during this month. And by 2023, this sequencing market is supposed to, they are actually growing in around 18%. They are supposed to reach $75 billion industry. Okay, whenever I can watch that out of 75, 50% is actually wet lab. Remaining 50% goes for the sequence analyzing platforms that are actually struggling to take meaningful data out of the NGS sequences. Okay, that's why we thought whenever you give a sample to a customer, it is a continuous journey. Even the customer paid us 15,000 something for their sample, we are guiding them in sequence analysis. Many times we are doing the analysis for them. And what is their objective? Is the objective met? How to produce a high quality pictures that can be published in a publication. Even after publication, still proposal preparation, submission to various industries, we are full-fledgedly we are guiding them because we want them to come back. So whatever service they ask, we usually provide. So this time we have a separate division called Barcode Academics. Actually, this academics actually involved with bioinformatics and proposing various bioinformatics analysis and various academic courses where we educate our customers and students, scholars in how to do this analysis. One of such program actually produced a research article where this author did not produce any wet lab data. They took the data from NCBI and they did this analysis. This paper has been published in Nature Scientific Reports. Okay, so yeah, they gave me an authorship also, the second author. All right. So like this, I really feel that the scope of bioinformatics is still in the improvement of the scope of next-gen sequencing, improvement of bioinformatics data pipeline, and the invention of robotics, especially for handling sample. Novasec and NextSec, they run for 2-3 days continuously to produce the required sequencing depth and length. So we require robotics in reagent handling during sequencing running, and new technologies in preparing the nucleic acid. I'm seeing this very clearly in last 20 years. the quantity of nucleotide, DNA, or the quality of DNA the quality has been increasing by years, and the quantity is getting decreasing in last 10 years. Okay, I feel in near future it will be scaled down to zeptoliters. Right now it is in microliter to nanoliter range. We can clearly see this is going to be in zeptoliters, and the machine size is being continuously decreased. Okay, there will come a time where Illumina real-time sequencing can be seen in the point of care applications or in the field, in a medical, agriculture, ecological application. Immediately, if you go just take the sample, real-time data, you can see. This is already happened then and there, but consistently as a product, it is not available, but it will be in near future. So when these technologies are continuously evolving, I really feel that bioinformatics data pipeline optimization is very much important, and there is a big scope for people who are skilled in bioinformatics or next gen sequence analysis. Okay, I know we actually created many of the startups, 2 to 3 startups now, who are actually supporting us from outside for next gen sequence analysis. Okay, many of these guys just don't have any office by physical space. Okay, they have just a laptop, they have a AWS service, they will log into it maybe in a cafe coffee day, just sit, sip a coffee for 3 4 hours, do the analysis using their Wi-Fi and get out. They are a freelancers, they are doing very very well. Okay, so many students run startups, or we have launched because of various programs we are actually giving in next gen sequencing services. So to conclude this webinar, I really feel that these three important areas which will be significantly increased because of the rate of evolution of next gen sequencing services and their sequence analysis technology. Okay, these three area were human health, agriculture, and environmental conservation and beyond. This will be a really three main area that you will see many more applications will happen. So the actual one more reason behind this webinar is to introduce you to the program that we are actually doing in Barcode Academy. This program is going to happen in a week's time. November 7th, it will start. It's a 7-day program where we will train the participant in next gen sequence analysis. Okay, the first day will be all about the introduction of background about the next gen, how their samples are treated in the company, what is the wet lab principle behind preparing sample for next gen. The second day will be fully about the principles of how the sequencing is done. Even though we did not see, we have saw about various platforms, but we did not see the exact mechanism. We will talk about how it is happening. If possible, real-time data and examples will be given. Third day is all about sequence analysis. We will initiate you into sequence analysis where Linux Ubuntu is the main. You will see graphical interface systems will not be that useful when it comes to analyzing next-gen DNA sequence data, which will be in GBs. So we will introduce you to Linux platform. Even though you have a Windows, there is a way to have a parallel server, Linux in your system, and you can learn next-gen out of it. So fourth day, we will train you in sequence, running the codes for sequence analysis, how to filter out your raw machine output file, how to clean the primer sequence out of it, how to do a multiple sequence alignment. Okay, we will have the basic bioinformatics analysis of the next-gen sequences using real data, and how to count the OTUs. That will be a sixth day objective, feature count, OTUs, operational taxonomic units. And after doing all the analysis on the 7th day, as one of the application, we will see a heat map analysis or the taxonomical analysis by green genes and NCBI. We can take many more reference sequences. We can merge it with our sequences to know their taxonomical position and many more. Okay, so this is the objective of this 7-day program. If you are interested, I have already shared this poster in the group, in the WhatsApp group. If you need this poster again, you can take it from the group, or you can take a screenshot of this slide, the QR here. You can scan, you will be prompted with a Google form. You can finish it. Yeah, there is a small fee to it, around 1,850 for our entire 7-day program. You will be gaining the skills on next-gen sequence analysis. If you are planning to do next-gen DNA sequencing, I'm very sure that this workshop will be of much use. You can have your real data. When you have your real data, you'll know what to do from A to Z, what are other things to be included in the pipeline of the analysis. Along with that, you will be given a e-certification for completing the course. So if you are interested, do register for this program. And thank you very much for joining this webinar. Every attendee of this webinar will be given a e-certificate. There is a link for downloading your e-certificate that I will share today, maybe before 11 a.m., 11 p.m. today. So there will be an Excel sheet containing your name and certification number. Use the number to download your certificate in the link I will be posting in the WhatsApp group. Okay. So if you want to celebrate your this next gen, whatever small information we can give it to you, please do so in the social media. I will give our social media links. If you want to tag us, please do tag us. So if you have any questions or any suggestions, feel free to comment it in the YouTube link. I will personally see to answer each and every of your questions, even during the webinar. We will do it. We will be answering your questions in the comments. Okay. Thank you for joining. Have a nice day.